I'm going to talk about how translational control and energy production are actually orchestrated in the cell. And I mean, I was trained after med school as a biophysicist, biochemist. So then I stumbled into this very complicated world of systems biology that I still don't understand. But I was grateful enough to have a very good friend who's a bioinformatician, so he helps me sort of cruise to this world of systems biology, and I hope I'm going to die before him, because if he dies before me, I'll have to stop doing research. So this is how what's going on? Okay. So this is how you know everybody starts to do science. I was in medical school, and then I started doing some molecular biology, and basically. As everybody else, I was observing these complex biological systems and trying to apply this knowledge to either disease states such as cancer and metabolic syndrome, or physiological processes such as learning and memory. But I used to spend my summers in the place called Korčula. This is the south side of Korčula. My grandparents had a house. And then I moved to a place that looks like this, which is Montreal in winter. So then I had to use my limited intellectual capacity actually to figure out how to survive, because for me this was really shocking. And then I figured out that I actually have something in common with the things I study. And if you position yourself into perspective of a cell, so cell doesn't see all these complex questions and whatnot. The only question that it actually has to answer is how to survive. And so when the cell is stressed, what happens is you have activation of a bunch of signaling pathways. This leads to changes in gene expression, and cell either mounts optimal response and lives, or it dies. So this is where my interest in how stress is involved in regulation of gene expression came to being. So as you will appreciate, the gene expression has been studied for a long time, but the vast majority of the efforts actually was focused at the level of transcription, whereby it was postulated that the transcriptomes are linearly translated into proteomes. But now we know, thanks to some work that was done by people, that there is a very strong RNA consortium here in Barcelona, so I'm pretty sure you're familiar with this. So it turns out that steady-state mRNA levels of what we refer to as transcriptomes do not actually perfectly correlate to what's happening at the level of proteome. And depending on the organism, who you ask, this correlation is going to be less or more perfect. But the fact is that it's not absolutely perfect. Actually told us that there is another layer of regulation of gene expression that's positioned between transcriptomes and proteomes, and this name was given by Jack Keane, Gorospe, and others. So this layer was called ribonome. And what happens in ribonomes, in these messenger ribonucleoprotein complexes, you have interaction of cis RNA elements that are located mostly in the UTRs, so the RNAs, with transacting regulatory factors such as RNA binding proteins and non-coding RNAs. And these are very dynamic structures in which fate of mRNA is sort of decided. And they play a major role in shaping of the proteins. So then if you go a bit back to 1620, this guy, Francis Bacon, who basically was a forefather of a scientific method, recognized that if you don't sort of use precise semantics in applying scientific method, there are going to be problems. He called this idolaphori, or idols of the market. And basically, if you read the last sentence, it's very important to refer to the things as precisely as possible. So if steady-state RNA levels equal gene expression profiles, and gene expression is the process by which information for a gene is used in the synthesis of a functional gene product, usually a protein, and if we know now the steady-state mRNA levels of transcriptomes are not reflected perfectly in the proteomes, then we shouldn't call steady-state mRNA levels gene expression profiles. And this was recently measured by many, including Matthias Selbach's group. And as you can see, protein translation plays a major role in regulation of gene expression, as well as other, such as other post-transcriptional mechanisms for regulation of gene expression, such as such as mRNA stability, also protein stability, which is obviously post-translational, plays a role. And my major interest in this process is because all the post-transcriptional programs are actually coordinated at this level. And as you can see, this was published in Nature, so it seems that Nature editors don't care much about semantics as well, because as it happens, protein translation doesn't exist, because mRNA gets translated and proteins get synthesized. But I guess nobody cares about Francis Bacon anymore, because this was long time ago. So translation, as you're all familiar with, there were talks by Nachum Sonnenberg and Tommy Lane here, so I'm pretty sure they explained this to you quite a bit. So it takes place in three distinct phases: initiation, elongation, termination. And initiation is best worked out, and we think that most of regulation of translation happens at the level of initiation. So I'm at the Department of Oncology, so my interest is mainly when it comes to pathological states. My interest is mainly in cancer. So although Bob Weinberg didn't put translation in hallmarks of cancer, there is actually a long-standing evidence. This is obviously not the original figure from this Italian pathologist called Giancarlo Pianese, who in 1896 observed that nucleoli in malignant cells are irregularly shaped and bigger than in normal cells. And of course, at the time, he had no idea what was going on in nucleoli, but now we know that nucleolus is a place where you make ribosomes, and this directly links ribosomes to ribosome biogenesis and translation to cancer. And as you can see, the year is 1896, so this is actually before Warburg came with his hypothesis. And then we also have a lot of evidence that a lot of translation initiation factors their expression is either decreased or increased in cancer. And the particular interest of my work in both Kathy Gordon's lab and Nachum Sonnenberg's lab was this protein that was discovered by Nachum Sonnenberg, that is eukaryotic initiation factor 4E, that binds to the 5' mRNA cap structure and regulates initiation of translation by rerouting ribosome to the mRNA. So you can see, and there was a lot of work done here by Santiago's group that shows that expression, phosphorylation and phosphorylation of 4E are dysregulated in cancer, and he has some more exciting data that we hope is going to come out soon. And also the 4EBP proteins, that are inhibitory proteins that bind and inhibit 4E's activity, happen to be decreased or increased in expression, and also there is increased phosphorylation of 4EBP1 in these tumors. So the major pathway that regulates mRNA translation, and some people would say that it's a center of the world, is this mammalian target of rapamycin pathway. So it's very complicated, and I've been working on it quite a bit, and I still don't understand half of it. So I won't get much into the detail, but mTOR exists in two distinct complexes. One is mTORC1 and the other one is mTOR complex 2. So they have different components, whereby Raptor defines mTORC1 and Richter defines mTORC2. And they're also functionally different, and they show different sensitivity to inhibitors, where this allosteric inhibitor, rapamycin, actually prefers to inhibit mTORC1, and then in some cells and after prolonged treatment, in some cell types and some organs like liver, turns out to inhibit mTORC2. So mTORC1 is actually a major regulator of mRNA translation, and two major targets are 4EBPs that were discovered by Armin Pause in Nachum Sonnenberg's lab and Jeffrey Lawrence at the same time, and the acid kinases that were actually discovered by this lady here in the first row here, Sarah Kozma and George Thomas. And these two are major targets that mediate the effects of mTORC1 on translation. So our major interest is on 4EBPs, and what 4EBPs do, they bind to EF4E and inhibit its activity. There are three 4EBPs in mammals, 4EBP1, 2, and 3. Once you stimulate the cells with hormones or growth factors or nutrients, what happens is mTORC1 gets activated, it phosphorylates and inactivates 4EBPs, which leads to dissociation of 4E from 4EBPs, and this allows 4E to bind to this large scaffolding protein 4G and to recruit this helicase EF4A, which actually presents the formation of the EF4F pre-initiation complex, and this complex actually enables scanning of the 5' UTR by the smaller ribosomal subunit. So signaling to AF4E is actually disrupted in cancer, and here I'm pretty sure you're familiar with all these mutations that occur quite frequently in cancer. So either activating mutations in receptor tyrosine kinases or in F kinase pathway will actually activate phosphorylation of 4E, whereas loss of all these tumor suppressors such as LQB1, tuberous sclerosis complex, and P10 will actually lead to hyperactivation of mTORC1 and inhibition of 4EBPs. So in the middle of this whole shebang is actually mTORC1, and that's where our interest comes as it being a central regulator of protein synthesis. So does this hold in the real world? Yes, and actually the pioneer evidence came from Santiago's lab actually, where he showed that both the levels in phosphorylation of 4EBP1 actually increase when the grade or severity of the breast cancer rises. And basically what this is a bit counterintuitive, that the levels of 4EBP will go up, but you have to remember that this phosphorylation will actually inactivate them. So it seems as if cell tries to compensate for increased activity of 4E, but by increasing the levels of 4EBP1, but tell us the signaling pathways give up, and then you have a phosphorylation of 4EBP1 and its inactivation. And this is one of the most and the best reproduced works in amateur field. So Santiago did very well. And more recently, Tommy Lane, who gave a talk yesterday, actually showed that if you treat with this new generation of active site inhibitors who, unlike rapamycin, target the active site of the kinase and equally inhibit mTOR complex 1 and mTOR complex 2, is actually determined by the ratio of 4E and 4EBP, whereby if you have cells that don't have any 4EBPs, the activity of active site inhibitors is almost absent in vivo, and then once you knock down 4E, you increase their activity. So it seems that 4EBPs are major mediators of the oncogenic effects of mTORC1 signaling. So another thing that interests me outside of translation is actually how gene expression is coordinated, and in particular how it is coordinated in stress. And why this is important is if you look at the typical study when somebody studies control of gene expression, what you do, you look at a very limited number of factors and then you blame everything on it's like when the reviewer asks you if you overexpress BCL2, whether you're going to rescue your cells. Of course you're going to rescue your cells. This doesn't mean that BCL2 lies downstream of whatever your pathway is. So if you have a musician, although he can be very good, as Stefan Grappelli here, when they play a tune, it actually has to be transcribed into a harmony that symphonic orchestra is going to play, which means that the notes this guy plays won't be the same as all these people play, but you're going to perceive it as the same thing, as the same tune, because the harmonies are going to produce the exact same tones. So this goes with orchestration of a tune, but also our brains seem to be built the same way, because this is an experiment that was done extensively in 1920s by Gestalt psychologists, and they used to show this diagram to people and ask them what this could be. So if you study control and you say, okay, we're going to come up with small pieces of a puzzle, and then somebody very smart is going to come and put this puzzle together, actually this is a very hard thing to do, because if you look at this thing, it's very hard to say what it is. If you look at the full picture, you will actually be able to see what it is, and it's an object that everybody can recognize, and then it's actually going to be much easier to break this into its parts and then figure out whether this or that would be a better cancer target. So to study this, we actually look at the gene expression at a genome-wide level, but we also look at multiple layers of gene expression. So we look at total, then cytosolic mRNA, and then we also isolate translationally active mRNAs that are associated with the polysomes. We analyze this. This slide is pretty old now. We do RNA sequencing. We used to do microarrays. Although we still compare the data from microarrays and RNA sequencing, there are some issues we can discuss. And then we basically compare the mechanisms at which level the changes in gene expression occur at the global level when it comes to specific functionally related genes. So the problem with this approach was always that you look at the differences in gene expression after, I don't know, 2 to 12 hours or so, let's say 2 to 16 hours, and then your physiological output is proliferation or survival, which usually takes more than that. So you have this lag period that sort of complicates the analysis of the results, because you just approximate what happens between, you see changes in gene expression and when you see what happens at the physiological level. So to overcome this, we started to look at metabolome, and the reason why is because the changes in protein actually take seconds to be reflected in metabolome. And so if you look at all these low molecular weight organic and inorganic compounds that are products of intermediate metabolism, it takes only seconds to detect them when you have changes in protein. And for this reason, we can simultaneously capture the changes in metabolome and in gene expression networks, and then try to tease out the gene expression modules that actually coordinate changes in metabolome. As you can see here, for example, normally if you want to study energy stress, you stress your cells for 32 minutes to 12 hours. You look at your changes in gene expression. After that period of time, then you need about 24 hours to see first BRDO positive cell, because they have to enter the S phase. So you actually overcome this problem by looking at metabolome. And then again, metabolism is playing a central role in cancer, which was reinvented now, but original study, of course, comes from Otto Warburg. And basically, there are profound metabolic changes in energy metabolism in cancer cell compared to normal cell that are collectively referred to as Warburg effect. So it also turns out that protein synthesis is the most energy consuming process in the cell, where it consumes about 20.5% of the ATP. And you have to add to that the RNA synthesis, because most of the RNA synthesis is actually making ribosomes. So both ribosome biogenesis and translating the mRNAs takes a lot of energy out of the cell. So the question obviously was, so how does cell know how much energy to make to fuel protein synthesis, and vice versa? When protein synthesis is inhibited, how does cell know not to put too much pressure on its mitochondria to make ATP? So to do this and to actually it was a great opportunity to study how gene expression is coordinated rather than regulated. And so what we looked at is we took three inhibitors that have very different mechanisms of action that all impinge on mTORC1. So rapamycin is allosteric inhibitor of mTORC1. It doesn't affect mTORC2, and it inhibits acid kinases a bit better than 4-EBPs. Inhibition of 4-EBPs is not complete. Active site inhibitors inhibit both mTORC1 and mTORC2, and they inhibit all known mTORC1 outputs to the same extent. And then metformin actually gets to mitochondria and then interferes with complex 1, which leads to increase in MP-ATP ratio, activation of LKB1, that leads to activation of MPK and inhibition of mTORC1 signaling. As Adem and Anand showed this in their cell metabolism paper in George Thomas's lab, and Sarah Cosmo's lab actually, is that metformin can also affect mTORC1 directly through these small Gtpases called RAGs. So when we did this exercise, we wanted to be very stringent, because when you run large-scale data analysis and you only come with new things, then it's very hard to say whether they're new or false positive, regardless what your FDRs and p-values are. So we basically looked at the overlap and of all three drugs, and we found that 65 messenger RNAs are actually downregulated at the FDR less of 0.01%. And then we validated these en masse on using an NanoString technology. So instead of running a couple of real-time PCRs, we actually validated about 80 genes. So we have representative genes here presented. And what came up is, so at the same time, Sabatini and Ruggiero's lab ran the same study. So they've seen 5' top mRNAs that go into this 12%. So they do represent a large number of mRNAs, as we all know, that are regulated by mTORC1. But what struck us is that actually RNAs that are transcribed, that are synthesized in the nucleus and encode mitochondria-related RNAs for 15% of this 65 RNAs. So basically they were highly enriched in this category, and we used gauge analysis to come up with this. So then this sort of brought us to our main question, which is how are translation and energy production coordinated, and this study was carried out I just take credit for the stuff that other people have done. So it's carried as a PI. That's a very good thing you can do. This guy, Masahiro Morita, actually ran most of these experiments and really pushed this study all the way through. And then he held help from people in Valentin and Shelley in my lab, then Tommy Lane, who's sitting there in the middle, Mesley and Chadi in Sonnenberg's lab. We had some great help from our resident metabolome expert, Julie Saint-Pierre and Simon Pierre and Valérie in her lab. This is the guy I do bioinformatics with, who used to be a colleague of mine and Tommy's in Sonnenberg's lab. Now he's at Karolinska, and Christina in his lab. We have a Scottish connection. If you do metabolism, you have to have a Scottish connection. So we worked with Al Gottlieb as well, Liang Zhang in his lab, and then Jan in Arnim's lab, in Arnim Pause's lab at McGill, and then Michael Pollack is my long-standing clinical collaborator at Lady Davis Institute at McGill University. So when we looked a bit more closer to this mitochondrial RNAs, what happens is, so if you look at the genes, you see all these kind of genes, mitochondrial ribosomal proteins. They're sort of these mRNAs are abundant. You don't know whether they're there for a good reason. But we will notice that TFAM is there, which of course is a major regulator of mitochondrial biogenesis and function. And we also see components of ATP synthetase, or complex 5, and then complex 1 as well. And then when we looked at, when we subclassified the processes in mitochondria, it turns out that most of these mRNAs are involved in mitochondrial ATP synthesis. And then, we treated cells for 12 hours, so some people argue this is too long. You're going to see the effects in changes in transcription that will be reflected in steady state RNA levels. But we actually measured this directly. And as you can see here, if you compare the effects on the upstream mechanisms of regulation of gene expression versus translation, the magnitudes of effects on translation are much higher than the ones on the steady state mRNA levels. And what this told us that we can actually study the effects that are primarily translational after the 12-hour treatment. Why it was good for us to use 12-hour treatment, at the same time you can see effects on mitochondrial function, so we can again simultaneously monitor changes in gene expression with the corresponding functional outputs. And so, systems biology is great, but I think biochemistry is much better. And so Morita actually went and validated all these targets. And as you can see, 3 different active site inhibitors suppress expression of ATP5O, ATP5D, and TFAM at the protein level. COX4I1 is a component of a complex 3 that didn't change in our system, and we don't see any changes, so that was comforting as well. And then we did all the control plots to show that actually active site inhibitors will inhibit mTOR signaling. Then we looked at polysomes, and although there was, as expected, a decrease in formation of polysomes and increase in the monosome in cells treated with active site inhibitors, which actually shows you that global translation is inhibited, we could still clearly see polysomes, meaning that you still have ongoing translation of certain mRNAs. So we then looked at these 3 targets that we identified at the protein that were shifting at the protein level. Their RNA levels didn't change, but as you can see here, they shifted from heavy polysomes toward light polysomes and subpolysomal fractions, which shows that their translational efficacy was inhibited by these 2 active site inhibitors, whereas this wasn't the case for a beta-actin mRNA that's not mTOR or 40BP or 40 sensitive, and sort of we use this as a control. So basically what this with the genome-wide data showed us is that under these conditions, well, I should say in MCF7 cells where we looked as well, there is a selective effect of active site inhibitors on translation of this subset of mitochondrial mRNAs. So to link this to some functional data, we looked at mitochondrial function, and here you can see that both inhibitors suppressed strongly mitochondrial respiration. This wasn't, so metabolic organization stayed the same, meaning that coupled and uncoupled respiration was inhibited to the same extent. And then we could see that also mitochondrial biogenesis goes down because relative mitochondrial DNA went down, and the mitochondrial mass, as measured by mitotracker activity, went down as well. And finally, ATP amounts went down in the cell. And this is a pretty huge effect on ATP because people don't realize this, but cell does everything in their power to keep ATP levels constant. So rarely you see depletion of ATP of, I don't know, 80%. This is, if you see a data like that, this is probably some kind of artifact. So then we also looked at glycolysis, and glycolysis was inhibited as well. As you can see, glucose goes up, lactate goes down, and then the pyruvate derived from glucose went down as well. So this was all in steady state, so you don't know what's really going on. So to tease these things out, we actually did a flux analysis, and this is just a schematic showing you how we do it. So basically, we label carbon in glucose, and then this glucose runs through glycolysis, TCA cycle, and then in the first round, you're going to have citrate and isocitrate that are M+2. In alpha-ketoglutarate, there's going to be M+2, and then in the second round, you're going to start getting M+4 metabolites. So when we looked at this, it actually corresponded to the steady state levels of TCA intermediates, whereby active site inhibitors inhibit flux of glucose towards citrate, isocitrate, and alpha-ketoglutarate. As you can see, with the increase in M+0 and the decrease in M+2 and M+4 in all cases. So basically what this told us is that yes, active site inhibitors suppress translation of these mitochondria-related RNAs that are encoded in the nucleus, and this leads to inhibition of mitochondrial function and related metabolic outputs. So then we wanted to see whether which mTOR complex mediates these effects. To do this, we did something that's pretty much standard in the field. We knocked down, and by we I mean Tommy Lane, knocked down Raptor and Richter in these MCF7 cells, and you can see the knockdowns work. And then what was supposed to happen in the signaling happened. Only Raptor knockdown led to downregulation of ATP5O and TFAM, as you can see here, compared to the Richter knockdown that didn't have appreciable effect. What sort of perplexed us, what happened at the level of respiration, knockdown of Raptor, inhibition of mTORC1 led to reduction in mitochondrial respiration, but surprisingly, knockdown of Richter led actually to activation of mitochondrial respiration. We thought that the explanation is quite simple, that basically when you get rid of Richter, you have more mTORC1 signaling, but then Michael Hall saved us, and he showed why this is happening is much more complicated than just having more mTORC1. So mTORC2 complex also plays a role in mitochondrial respiration, but it's quite different, and it has to do with mitochondria and the plasmatic reticulum membrane and the flux of calcium and lipids that I don't really have time to get in, but we can discuss that later. So basically, respiration rates also went down, and then this was, again, both coupled and uncoupled respiration were affected to the same extent. And then, again, we looked at relative mitochondrial DNA. It went down with Raptor. It didn't go up with Richter or down. And then the ATP amounts didn't change with Richter, and it actually, they went down with Raptor. And then all the TCA metabolites except alpha-ketoglutarate changed only were suppressed only in Raptor knockdown cells. It's worthwhile noting that this alpha-ketoglutarate in MCF7 cells the carbon doesn't seem to come from glucose, because it's not labeled very efficiently, and we think that it might actually come from glutamine, and this would explain why because it's been shown that mTORC2 regulates glutamine-related metabolism, and that's why we think that we observe this effect in alpha-ketoglutarate. So just to make sure that it's mTORC1, we also took advantage of TSC knockout. So TSC is a Gtpase activating protein for RAB. And this RAB is RAS homolog enriched in brain that, when bound to GTP, activates mTORC1. So basically, TSC2 inactivates RAB, which in turn inactivates mTORC1. So the cells that don't have TSC show higher mTOR activity, as you can see on the lower panels. And then we looked at ATP5D and TFAM. And as you can see, the TSC2 knockout maps that have increased mTOR activity actually upregulate ATP5D protein and TFAM as well. We looked at the polysome TSC knockout. The polysomes TSC knockout have a bit more polysome. This was reported before, so there was no surprise there. But what we could observe is that ATP5D and TFAM actually get loaded more efficiently on polysomes. You can see they're shifting from these fractions to those fractions in both cases, but not beta-actin, that again showed us that in these cells you have selective effects of mTORC1 signaling on these mitochondria-related mRNAs. And then we looked at the respiration rates, and again, the mitochondrial DNA content and ATP amount, and they were uniformly up in TSC knockout maps as compared to wild-type maps. Again, demonstrating that in cells where you have high mTOR signaling, you have higher expression of these mitochondria-related mRNAs that translates into increased mitochondrial activity. So then we wanted to see which downstream target of mTORC1 mediates these effects. So there was some evidence in the literature that in lower organisms, 40BPs actually control mitochondrial biogenesis. So we started with 40BPs. And you can see that when you knock down 40BP1 and 2 in these MCF7 cells, you have rescue of the levels of ATP5 and TFAM in cells that are treated with active site TOR inhibitors. So just to remind you, once you inhibit mTOR, you actually activate 40BPs, which in turn inactivates 4E. So this actually made perfect sense. We looked at polysomes again, and this effect seemed to be translational. As if you look in the control cells, active site TOR inhibitors shift ATP5 and TFAM messengers, but they don't they don't have a major effect on beta actin. Whereas in 40BP1-2 double knockdown cells, this effect is absent, and you don't have any appreciable shifts in ATP50 and TFAM. So this simple biochemical experiment actually showed us that 40BPs mediate the effects of mTORC1 on the translation of these messenger RNAs that are involved, that encode proteins that are involved in mitochondrial biogenesis and function. So again, we wanted to see whether knocking down BPs will actually rescue the mitochondrial phenotype. And it partially does. As you can see, there is a significant rescue in respiration, again, both coupled and uncoupled. And then there is also rescue in TCA intermediates, as measured by the flux of glucose to citrate, isocitrate, and alpha-ketoglutarate. So then we also looked at relative mitochondrial DNA, and shockingly, this was almost completely rescued. And the same goes to relative mitochondrial mass and the ATP amounts. So this basically told us that 40BPs do mediate the effects of mTORC1, but mostly on mitochondrial biogenesis, because the rescue of these TCA intermediates wasn't complete. And not all of TCA metabolism happens in mitochondria. People tend to forget this, but so we had to remind the reviewers of that fact more than once. But they made us do a lot of extra experiments as well. So then we basically looked whether TFAM that doesn't contain 5' UTR and it's not translationally regulated will rescue these effects. And this is again a thing that the reviewer asked for, and this comes from this control versus coordination thing. Of course, the TFAM is going to rescue the. So I won't look too much into this experiment because this TFAM construct doesn't have 5' UTR, so active site TOR inhibitors won't inhibit its expression, because they cannot regulate its translation. Most of the translational regulatory elements are in 5' UTR, but we here empirically show this as well. And then no wonder that relative mitochondrial DNA content and then increased in these cells, and then this was resistant to effects of active site TOR inhibitors. Again, I wouldn't, these kind of experiments are like expressing BCL2 and showing that your cells don't die. So to my knowledge, TFAM doesn't do anything else in the cell than to increase relative mitochondrial DNA content. And mitochondrial biogenesis. So this is a very expected result. One thing that bothered us, and actually reviewers pointed this out, I would say quite well. So we were very thankful for this. And this is a role of mitophagy, because mitophagy plays a major role in regulating mitochondrial function and number actually during stress. So the way Morita looked at this is he knocked down ATG5, which is a major regulator of autophagy, in wild-type mass and 40p double knockout mass. And this was probably done by Tommy Lane, if I think. Yeah. No, Morita. Okay, Morita knocked this down. And then, as you can see, by monitoring lipidation of LC3, when you treat with active site TOR inhibitor, you induce autophagy in cells where ATG in the control cells, and this effect is absent in ATG knockdown cells, regardless what their 40BP status is. And this is just to show that the inhibitor worked. And then we looked at the relative mitochondrial DNA content, and what happened is when you knock down ATG5, so just the knockdown of ATG5 actually rescues a bit mitochondrial DNA content, but when combined with a knockout of 40 BPs, it actually leads to a complete rescue. And this also goes to relative mitochondrial mass as measured by mitotracker, but mitotracker is not the best way to look at mitochondria. I happen to like electron microscopy quite a bit, and then this is the reason why, because here you can actually see appreciable effect where in wild-type maps you have a strong fusion of mitochondria that happens to be a defense mechanism of mitochondria to induction of their degradation by mitophagy, whereas this effect is completely absent in 40BP double knockout maps. So taken together, these data showed us that in cell lines, and we did this in a couple of cell lines, I showed you data mostly from MCF7s and MAFs, that mTORC1 actually inhibits translation of mitochondria-related mRNAs through 40 BPs, and this has a profound effect on both mitochondrial morphogenesis, biogenesis, and function. And then, of course, you have to do some stuff in mice to get published these days. So then we monitored the mitochondrial function in vivo. So these, of course, are not very precise measurements of mitochondrial function. But anyhow, Morita could see that active site, that if you treat mice that don't have 40 BPs with active site or inhibitors, you don't affect their oxygen consumption, locomotor activity, or heat production, which are all measurements of mitochondrial activity in the cells. And of course, the most important thing for publishing papers nowadays is having a doodle at the end that you call a mechanism. So this is our mechanism, and it actually shows that mTORC1, which we all knew, inhibits 40 BPs, which leads to activation of 4E. But what we show, what I think we shown here, is that what happens then is that 4E activist translation of subset of these mitochondrial RNAs, and all the increased translation and ATP consumption, you concomitantly increase ATP production by making more mitochondria that have higher activity, and this actually fuels back to translation. So basically, translation dictates mitochondrial ATP production in this feed-forward mechanism whereby then mitochondria produces enough ATP to get translation going as the most energy-consuming process in a cell. But what this also tells you is that what we shown before is that what also happens is all coordinated with proliferation rates, so it sort of makes a lot of sense that the translation and of course proliferation will increase ATP consumption, but this would be directly followed by increasing mitochondrial ATP production, which will fuel both translation and proliferation. This also has implications to when we inhibit mTOR signaling, because it's widely believed that when you inhibit mTOR signaling, you induce energy crisis in the cells, because mTOR signaling is a major regulator for anabolic processes and whatnot. And it seems, I mean, more and more data shows that it seems to be adaptory mechanism, and this sort of effect can in part explain it. So once you inhibit mTOR signaling, you inhibit mitochondrial ATP production, which could arguably cause the energy crisis. But the problem is at the same time you're going to inhibit ATP consumption. So what's going to happen is the cell is just going to go to a lower energy equilibrium and become metabolically dormant, and it's very hard to kill metabolically dormant cells. So this also tells you that once you target mTOR, the window of opportunity that you have to hit is also important, not just which patients to treat and so on. And so this would more or less be it. And I would just like to thank again the members of the lab that have to put up with me every day, although I'm away most of the time. So Valentina and Shannon, they started with me, and then I have a very talented PhD student, Arjuna, and then also postdoc Laura. This is a half of a master's student that's at the Department of Mathematics that I co-supervise, that helps us understand what, she translates what all us people do to us so that we can understand. And I see Morita as a junk member of the lab, because you know, he did most of this work, but he's physically in Sonnenberg's lab and he's looking for a job, and this guy is really good. And of course collaborators, so Ola does all the bioinformatics. As I said, without this guy I wouldn't be able to do any science. So thankfully enough, he's younger than me, and he's Swedish people I heard live long, so I guess he's going to live longer than me, so which is a good thing. And then Tommy Lane, who was here and who just got a position in Ottawa, and then I mean this guy did pretty much every overexpression and knockdown when I was in Sonnenberg's lab, and much more than that, and never took any credit for it, which always annoyed me. But finally he has his own place, so I hope that he learned the hard way that he shouldn't be nice to the people that are not nice. And then of course Nahum Sonnenberg, who was a great mentor to me. I mean, I spent 2 years in his lab and I learned more than what I learned in science combined before and after that. Michael Pollack was a long-standing clinical collaborator, and then Julie Sapierre, who helped us with metabolome studies. And then I would just like to acknowledge people that give us the money. So these are the local Canadian and Quebec associations. This money comes from the United States, and this, believe it or not, comes from Sweden. So we got some money from Swedish taxpayers, which is a pretty good thing when success rates are 12% in Canada, and I don't know, 7% in the United States. And thanks for coming here and listening to all this stuff. I hope it wasn't too boring.